You just tell me when you want me to start.
Yeah, I will. I'm waiting for her to give us a sign. Oh. Okay, I welcome you all to the second live webinar of the Department of İstanbul Arel University Molecular Biology and Genetics, organized in collaboration with the Gene Arel Student Club. The subject of today's webinar is gene therapy. As everybody knows it very well, gene therapy is a medical approach that treats or prevents diseases by correcting underlying genetic problem. Today, people with genetic diseases count on gene therapy as a powerful cure the disease permanently. However, there are still concerns that we are not sure about it yet. For example, cost efficacy is a major issue since it is very expensive, especially for the developing countries. And safety in terms of genotoxicity is another major drawback of gene therapy. And immune response against the vector-delivered gene is a potential risk. Despite all these drawbacks, gene therapy is still the best option to cure for every genetic diseases. Today, we have a distinguished scientist who has been working on gene therapy in respiratory diseases from Imperial College London. He's going to catch us up with the recent updates in gene therapy. I appreciate him very much for accepting our invitation to give a talk. I will cut it short because I'm sure everybody cannot wait to listen to him. Now I shut up and let Nazlı, who is a member of Gene Arel Student Club, to introduce Dr. Eric. Please, Nazlı, go ahead.
Yes, hocam.
I think you're on mute.
Sorry, Nazlı will introduce you.
Ses gelmiyor sanıyorum, Nazlı'nın sesi kapalı.
Biliyorum hocam.
She's on mute.
Yeah, she will talk from here.
Hello.
Hi.
I also welcome you all to our live webinar. Thank you for attending. And I want to introduce Professor Eric Alton. Eric is a professor of gene therapy and respiratory medicine at Imperial College, London, an honorary consultant physician at the Royal Brampton Hospital and IHR senior investigator. Over the last 20 years, he has coordinated the UK Cystic Fibrosis Gene Therapy Consortium, which brings together all the key centers in the UK in a program to deliver gene therapy for these patients. Based on the cystic fibrosis program, the consortium is now also applying the underpinning gene delivery technology to a number of other diseases. He is the chair of the NIHR ATMP coordinating group, which has pulled together a dozen of the key stakeholders in the field to catalyze coordination and avoid duplication. He is the lead for Cluster B of the NIHR CRN, coordinating portfolio studies in respiratory, ENT, gastroenterology, hepatology, and infection. So, Dr. Alton, please go ahead. Thank you.
Thank you very much indeed, and thank you for the very kind invitation to speak. I was just saying earlier, it would be a real pleasure for me not to speak for 40 minutes without interruption, and I would be really, really pleased if you would ask questions and heckle me and throw things because that would make the whole talk much easier. So I'd really appreciate it if that was possible. Just as I say, by background, I'm a doctor, but I only see patients once a week. So people say I'm not a proper doctor. I try and do some science, but I'm medically trained. So people say I'm not a proper scientist. So somewhere in between the two. So what I'm going to talk to you about is an update on gene therapy, as you see, for respiratory diseases. But all of you will know that the main focus has been in cystic fibrosis. So I'm going to largely tell you about our work on cystic fibrosis, but I will also talk about other respiratory diseases. Now, I'm sure you are aware the cystic fibrosis gene was now cloned more than 30 years ago today. And I'm just going to update you, first of all, on what is the problem that we are trying to treat with this fancy new therapy, with gene therapy. And secondly, where have we got to? On occasions, I'm going to mention this gentleman to you, and I just want to indicate to you a very clear conflict of interest. Now, I don't know whether you're all world experts on cystic fibrosis or not, but just in case not, just to briefly go through what we're trying to treat, it's the commonest fatal condition in the Western world. In the audience, 1 in 20 of you will be carriers because this is a recessive condition and therefore carriers are happy and healthy. The problem, of course, comes when 1 in 20 carrier gets together with another 1 in 20 carrier. And the chances of that, of course, are 1 in 400. And if you have 4 hypothetical children, then 1 in 1,600 live births should be children with CF. It turns out to be about 1 in 2,000. And it's fatal because the disease focuses on the lungs with the build-up of thick, sticky secretions. And there's nothing that bacteria like better than thick, sticky secretions. And these unfortunate patients get repeated chest infections, and the median life survival at the moment is just under 40 years of age. So one of the key questions is, how do you get from the mutation, the genetic mutation, to the death of these patients from respiratory infections? So if we take a trip down the airways, and here is the trachea, here are the bronchi, the bronchioles, and the next picture will show you one of these bronchioles cut across. And just imagine you are floating in the middle of the bronchiole here, looking down on the surface epithelium. So here is now the airway epithelium up here. This is the cytoplasm. Here is the cell membrane. Here is the cytoplasm. And this is the protein the cystic fibrosis gene makes, so-called CFTR. CF for obvious reasons. T stands for transmembrane because this protein appears to span the membrane and is shaped like a pore. It, excuse me, it looks as if this pore should transmit something. And R for this regulatory or golf ball-like structure, which appears to move in and out of this pore-like structure and regulate it. Now, that all sounded very academic and excellent. It's a load of old nonsense. The real reason this was called CFTR is the team in Toronto who cloned the gene, when they were doing their pipetting in the laboratory, used to listen to the local radio, and the local radio station is called CFTR 680, and they simply made the words fit the acronym. Now, CFTR does transmit something, and that something are chloride ions, and it moves chloride ions from inside the cell across the cytoplasm and onto the airway surface. And all of you will remember from your days of physics that if you move negatively charged ions, you are moving an electric current. And so you should be able to measure a voltage or a potential difference on the airway surface. And about 25 years ago, I put together a diagnostic test where we put a tube into people's noses, we measure the voltage, and we can say whether you do or do not have cystic fibrosis. And we now have pediatric and adult clinics at the Brompton Hospital using this technology to say whether you do or don't have CF. Now, you'll also remember from your biology days that every time you move an ion, it doesn't matter which ion it is, water must follow by osmosis. And so what all of you are doing listening to this talk is you are regulating the water volume up here on your airway surface. Now, one of the questions is why you should be interested in regulating your water volume. Well, here is what your airway surface looks like. The cell is underneath here, and here are the cilia, and here is the mucus that covers the airway surface. And all of you are inhaling at the moment bacteria in the rooms where you are sitting, and not in your clean country, but certainly in London, I'm inhaling all of this. And these bacteria and these pollutants will destroy the lungs if we don't get rid of them. What happens is they get trapped in the mucus, and the cilia flick the mucus and therefore the bacteria and the pollutants out into your throat, and you cough the material out or you swallow it. And this defense system called mucociliary clearance is an absolutely key defense system to defend your lungs. And one of the things that we are very clear about now is that these cilia have to stand in the optimal volume of water. And if there is too little water or too much water, then the cilia will not work properly. And I think you can now see what happens in the unfortunate CF patients with their gene mutation. Because there is no CFTR moving chloride here, there is no water. Therefore, the cilia flop over. They don't sit in the right volume of water. In come the bacteria. The cilia don't work properly, so the bacteria stay in the lung. They get repeated chest infections, and then, sadly, they perish from lung disease. So this is the most likely link between the genetic mutation and the death of these patients. And please think of CF as a genetic disease of lung water movement. I'll just stop there just to see if there are any questions on pathophysiology.
Any questions? Sorusu olan var mı? I think there's no question, but so the CFTR function is important here for the chloride movement. So, chloride movement or the export of chloride out of the membrane is a regular process, or?
Exactly. It's an absolutely sharply regulated process and is critical for all of us sitting listening to this talk. Without CFTR, we will get lung disease.
Mm-hmm. Okay, thank you.
It's obviously very easy to sort out this problem. All we have to do is a bit of gene therapy. All we have to do is replace the missing CFTR protein in the lungs. So in case any of you are enthusiastic enough after this talk to give us a hand, and we could always do with a hand, I'll just give you a quick do-it-yourself guide on how to do CF gene therapy. All you have to do is take out a cotton bud, which all of you will have brought to this talk. Just lean over to the person next to you and put the cotton bud inside their cheek. Rub off a cell from inside their cheek. Bring it along to the laboratory. We can focus down on the cell, on the nucleus. We can focus down on the long arm of chromosome 7, which is where the CF gene is. We can, of course, then cut the CF gene out here, and then put the CF gene into some E. coli bacteria. As the E. coli bacteria multiply up their own bacterial genes, they will, of course, multiply up the human CFTR gene you have put in the E. coli. After a few days, you can then mash up the E. coli bacteria, pour the contents into the nebulizer, turn on the gas, push the CFTR gene down into the lungs of the patients, and then you can go to Stockholm and pick up the Nobel Prize. This is very, very easy and straightforward.
So is it the way of gene delivery with the gene therapy?
Sorry?
Is it the way that you deliver the gene with this?
Correct, with the nebulizer. Oh, exactly.
Nice.
Now, along the way, I think you will find there are some difficulties that mean you may not end up in Stockholm for the Nobel Prize just yet. And so one of the questions is, there is this mucociliary clearance system which is trying to get rid of the bad things, the infection and the pollutants. Why would it not also get rid of the gene when you put it into the lungs. And of course it does that. Now, even if you could get over the mucociliary clearance, there is the respiratory epithelial cell underneath, which has got this formidable cell membrane, which is meant to keep things out. So even if you could now get the gene across the cell membrane, you've now got to get it across the cytoplasm towards the nucleus. And you will all know that there are endosomes here in the cytoplasm waiting to break down materials in the cytoplasm. And even if you could get the gene into the nucleus, you have to bear in mind that the nuclear pore complexes here in the nuclear membrane are too small to allow uncondensed DNA into the nucleus. And even if you can get the DNA into the nucleus, you have to persuade the nucleus, which is used to endogenous DNA, to now transcribe exogenous DNA that we have put in, and then to correctly translate it and put the CFTR protein where it should be on the cell membrane. So the reason that CF gene therapy is not easy to do is we are working against evolution. Evolution has made the lungs to keep things out, and we are trying to put something in. So if you are going to help us, please do not do that experiment. Don't put the CF gene in a nebulizer and turn it on, because it will not work. The gene will not go into the cell because of the evolutionary barriers. And what you have to do is you have to help the gene into the cell. And you only have two options. The first option is to use a virus. Why don't you put the CF gene inside a virus? The life ambition of any self-respecting virus is to give you a cold. It's to put its genes into your respiratory epithelial cells, in the same way, of course, as coronavirus does. So if you go home this evening and put the CF gene into a virus, you will be very pleased with the result. For the next 2 or 3 weeks, you will find that the CFTR protein is formed here on the surface. And then after 2 or 3 weeks, the protein will disappear. You only get a cold for a couple of weeks. You will only get CFTR protein being produced for a couple of weeks. And it will then occur to you that CF is a lifelong condition, and you have to do this gene therapy repeatedly. And this is where viruses go horribly wrong, because the body's immune system says, I have already seen that virus once, and I will mount an immune response to it. You mentioned that in your very kind introduction. And so if you want to write a brilliant Nature paper, use a virus, because it will be very efficient at producing the CFTR protein on the surface. If you are wearing a clinical hat, you will find that viruses are not a good way to go, because they cannot be repeatedly administered. So your only other option is to wrap the CF gene here in fat globules or liposomes. And the fat fuses with the fat of the cell membrane here, and it dumps the CF gene into the inside of the cell. This has been going about 20 years, and this has been going for about 20 million years. So this is going to be much more efficient, but cannot be done repeatedly. This is much less efficient, but can be done repeatedly. So where we were when we started this program is the only way to carry the CF gene into the target respiratory epithelium is to use a non-viral approach, to use the liposomes. So in response to this challenge, we brought together everybody in the United Kingdom who is working on CF gene therapy, and there are groups at Edinburgh University, Oxford University, and my own group at Imperial College. And this has focused the hearts and the minds and the souls of 50 clinicians and scientists in a milestone-driven common strategy. We share all our data, we share all our resources, we don't allow duplication or competition. We're probably not the best example of a democracy in the world, and we work very much more like a small pharmaceutical company than a group of academics. So what I'm going to do is just address the question of, over the last 21 years, we've been working together for 21 years now, where have we got to? So the first thing we did was to heed our own advice and to use the liposome to transfer the CF gene into the target epithelium. But one of the things that we said to the patients, and one of the things we said
Can I interrupt you here?
Please.
Yeah. So do you clone all the gene into liposome or viral vector, or just part of the gene?
I will show you a picture in a second.
Okay.
We put the whole CF gene in.
How big is the gene?
It's big. It's about 7 kb or so. And in liposomes, and also in the next bit of the talk I will show you, liposomes can take huge amounts of the genetic material, and we can put the whole CF gene in. It's a really good question.
Thank you.
So one of the things we explained to the patients and the funders is we were calling this wave 1, and that is a deliberate strategic decision to say to them that we do not believe liposomes will be the final answer. We do not believe, when we started this program, that the National Health Service will end up with a treatment for CF using liposomes. Now, this is the 67th in a series of 250 liposomes produced by our pharmaceutical partner Sanofi Genzyme. And in answer to your question, this is the 169th piece of plasmid DNA the consortium produced. From here to here is the complete CF gene. And these are the promoters and the enhancers that we were using. And if you're interested, we were using a human CMV enhancer and an elongation factor 1 alpha promoter, which we thought was the best combination. So we think if there is a What Gene Therapy magazine, this would be the best buy. After 10 years of preclinical work, we think this is the best combination. So we did, as I say, a decade of preclinical work. We outsourced the clinical manufacturing of this product across 2 different continents. We did regulatory compliant toxicology. We wrote kilograms and kilograms and kilograms of paperwork for the regulators. And we then undertook 6 phase 1/2A clinical trials where we checked for safety and we checked for proof of concept the CF gene was being expressed in the CF patients. And from there, we moved towards the world's largest phase 2B clinical trial. Now, I'm sure all of you will recognize the Kitty Hawk aeroplane from the Wright brothers in 1903. This plane did not fly every day, and when it flew, it only flew 50 yards. But it proved the concept that powered flight can happen. And so we explained to our patients before the clinical trial that our most optimistic hope was, for the first time in the world, if we gave the CF gene with a liposome every month for a year, that we could begin to show the concept that their lung function could improve. That was our most optimistic hope. So we are a very imaginative bunch of people, and we called this the multi-dose trial. It was double-blind and placebo-controlled, and the placebo was salt solution. And I think you could criticize us for using salt solution, and I'd be delighted to take any questions on that. We gave a dose of gene therapy once a month for a year, because this is a lifelong disease. We optimized beforehand the optimal volume of material we should nebulize into the lungs, and we found that was 5 milliliters. We only treated patients with an age of 12 or more, and that is because lungs, as I'm sure you know, stop growing at the age of 12. And if we were to do something terrible to these patients, I'd rather do something terrible to a fully grown lung rather than a developing lung. The entry criteria were that the baseline lung function, so-called FEV1, the amount of air you can blow out in 1 second, had to be better than 50% predicted, because if it was worse than 50%, the lungs would be full of mucus and we could not deliver the gene. and it had to be less than 90% predicted, because otherwise we would not be able to demonstrate benefit in the lung function. We randomized the patients 1-to-1 for either gene or placebo, and the clinical study was undertaken both in Scotland, in Edinburgh, and also at the Brompton Hospital. There were no safe... Please?
Sorry, may I interrupt you again?
Please.
So the lung function, does it depend on the type of the mutation?
Very good question. So there are, as you know, more than 2,000 different mutations in the CF gene. Some of them are worse than others. So yes, sometimes the lung function does depend, or the decline in lung function does depend on the type of mutation. Gene therapy should be agnostic to which mutation you have. It should treat all the 2,000 different mutations. So we did not pick and choose which mutation the patients had, because of course we are not repairing the faulty mutation. We're replacing and adding a new gene.
Thank you.
So there were no safety problems over a 1-year period. Here is the primary outcome of the study, which is FEV1, the amount of air you breathe out in 1 second. Above this dotted line is good, below this dotted line is bad, and here is the lung function every month that we measured for 1 year. There were 50-odd placebo patients, and they gradually reduced their lung function over the 1-year period. And in contrast, the treated patients with the gene therapy remained stable over the 1-year period, and the trial reached its outcome with a significant benefit of just under 4% in terms of lung function for these patients. We also measured thousands of other secondary outcomes, and I'm sure you're aware on this forest plot that anything that lies to the right of this dotted line, and these are things like inflammatory markers, imaging, lung function, and so on, if it lies to the right of the dotted line, it means it favours gene therapy, and if it lies to the left of the dotted line, it favours placebo. I think you can see that everything we measured favoured that there was a gene therapy effect. So overall, what I'm trying to say to you is we believe this is the world's first demonstration that CF gene therapy, when given every month for a year, can begin to prove the concept that the patient's lung function can improve. What I do not think this is this. I do not think that a 4% improvement in lung function is good enough to warrant the National Health Service, or whatever equivalent you have in Turkey, paying good money for this gene therapy. I do not believe that 4% is big enough change, because this is going to be expensive, as you mentioned at the beginning. Now, as I said to you, there are many things we have not anticipated in the world, but one of the things we anticipated was that we would need not just a wave 1, but a wave 2. Although it's always slightly worried us, it only seems to have one engine. So over the last 15 years, we have been working with an amazing biotech company in Japan called DNA-VEC. And we have been working on a virus called Sendai. And Sendai virus is in the room that you are all sitting in at the moment. You are all breathing in Sendai virus. And it is the most remarkable way of putting genes into the respiratory epithelium. So here is the airway of a ferret, and what we have done is put into the Sendai virus the gene beta-galactosidase, saying turn cells blue. And I think you can see that every single airway cell always turns blue. This is not the best example, this is the typical example. Sendai is the most fantastic way to do respiratory gene therapy. There is nothing better. Unfortunately, it is a virus. Viruses, as I said to you earlier, last 1 to 2 weeks, and you cannot repeat a virus. So while this is very, very exciting, and we wrote a Nature paper on this, it is completely useless for CF gene therapy because you cannot repeat it. Now, working with the Japanese biotech company, we worked out why Sendai was so good. And in the same way as coronavirus, it has spike proteins on the surface. And these spike proteins are called the F and the HN proteins. And the F protein docks with the receptor cholesterol, and the HN protein docks with the receptor sialic acid. And there happens to be both cholesterol and sialic acid receptors here on the apical surface of your respiratory epithelium. So the reason that Sendai is so brilliant is because it has the right proteins to dock onto the right receptors. Now, if we are talking about viruses, maybe we should at least talk about lentiviruses. I'm sure you're aware of the fact that lentiviruses, for example, HIV, is a lentivirus. And the advantage of lentiviruses is that they integrate into your own genome. So one of the advantages and one of the reasons we might talk about a lentivirus is it will give you long-lasting expression because it integrates into your own DNA. Now, lentiviruses typically look like this, and they have proteins on the surface where there are no receptors on the airway epithelium. So if you put the CF gene into a lentivirus and nebulize it into the lungs, nothing will happen because there is no receptor for any of these proteins on the lentivirus. So this is useless for CF gene therapy. Here is Sendai virus, which is brilliant, but is useless for CF gene therapy because it only lasts a week or two. So the obvious thing to do is, why don't we strip off these proteins on the surface of the lentivirus and replace them with these FHM proteins, and so produce what is called an FHN pseudotype lentivirus. In other words, this lentivirus was not born with the FHM proteins. We have artificially put the FHM proteins onto the lentivirus. And this is what we call our wave 2 product. So the obvious question is the wave 2 product better than the wave 1? So here is the mouse lung in vivo, and here is the log scale of gene expression measured by luciferase. Each individual dot is a mouse, and here are the control mice, so there is no expression. This is the material in the wave 1, the liposome, that produced a 4% change in patients. So we are clinically benchmarking our treatment against the liposomes that we used in the clinical trial. And in contrast, here is the lentivirus, which is clearly at least 2 log orders better than this 4% benefit that we got from the liposome. So one day it occurred to us that perhaps mice are not people, and maybe we'd better look at human tissues. So here are human air-liquid interface cultures. So this is pseudostratified respiratory epithelium. Here are the cilia, here is the mucus, and you are looking down on these air-liquid interface cultures in these Petri dishes. And what you are looking for is pixels of color to show gene transfer at these different time points. And you can't see anything because this is the control. This is the salt solution. Here are the liposomes that we use.
Sorry, your microphone is off. Somebody made your microphone off.
Can you now hear?
Yeah, we can hear you now.
Okay, where did you lose the talk?
Can you repeat the experiment from the control?
Okay, so here you are now looking down on Petri dishes containing these air-liquid interface cultures. You are looking for pixels of color, and this is the control with the salt solution, so you see nothing at the different time points. This is the liposome-based gene transfer that produced a 4% change in lung function in the patients, and I can just see some pixels of color here. And in contrast, the lentivirus produces hugely better gene transfer. So is it the wave 2 better than the wave 1? The answer is absolutely yes. It's at least 2 logs better. So the next question is, does the lentivirus, because it integrates into your genome, give you long-lived expression? So here you are looking down on the nose of a mouse which 2 years beforehand had a single drop of salt solution. The reason you can't see anything is because this is the control. This mouse, 2 years beforehand, had a single drop of the lentivirus expressing green fluorescent protein, and at the end of its lifespan, this is a mouse with a Zimmer frame at the end of its life, and it is still producing green fluorescent protein after a single dose of our lentivirus. Please beware that we are not claiming that a single dose of our virus will give you gene expression for the lifetime of a patient. This virus will hit the respiratory surface epithelium, and the lifespan of a respiratory surface epithelium is probably between 6 and 12 months. It can only give you expression for the lifetime of a patient if you hit a stem cell or a progenitor cell. The stem cells or the progenitor cells in the airways are extremely well hidden, and I do not believe that we will hit those with nebulized gene therapy. So whichever way you look at this, we will still need repeat dosing in human beings. And therefore, you have to ask yourself the question, is expression feasible with a virus on repeated administration? And I said to you at the beginning of the talk, viral vectors cannot be repeatedly administered. So we thought we knew what the answer was going to be. We took our Prozac and we did the experiment. Here is gene expression on a log scale. This is the amount of expression you get after 1 dose, and much to our amazement, this is what we got after 3 doses. So this is the only viral vector that can be repeatedly administered and still give you the equivalent of 1 dose. And there are at least 2 other groups in the world who are doing the same experiments in Iowa in America and in Adelaide in Australia, and they have the same data as we have. We don't yet know fully what the answer is, but the answer is that you can repeatedly administer a lentivirus into the lung. So this virus is completely unique. So the next question you ask yourself is, can it not just produce reporter genes, but can it produce functional CFTR protein? So this is a very clever assay, which I can say because we didn't set it up. This comes from the University of Utrecht. And what these people do is they take biopsies from the guts of CF patients and non-CF patients. And these organoids have non-CF cells lining the organoid here. Because they have functioning CFTR, chloride is moving water into the organoid, and the organoid swells with water because CFTR is working. If you do the same thing in CF patients, you will see that the organoid is lined with CF cells where the CFTR is not working, so no water, so no swelling. And we sent the team in Utrecht our virus, and we asked them to transfect these CF organoids, and you can restore gut swelling in these organoids with our virus. And we have many other examples of how this is working well. So there are some additional features of our virus that would support moving into the clinic as quickly as possible. And the first of those is it safe? Does it produce acute inflammation? So we are now in the mouse's lung. This is the material we used for a year in CF patients, and we saw no safety problems. And in the mouse's lung, the lentivirus does not produce any more acute inflammation than this lentivirus. So am I worried about acute inflammation in patients? Probably not. Secondly, this is an integrating viral vector, and it could produce cancers because it integrates into the wrong part of the genome. So here are 100 mice compared to 50 controls, where we have followed them for 2 years to see whether they develop lung or other tumours. And the answer is they have 100% survival. There is no difference compared to the controls. The next thing we did was we looked for hotspots for integration across the murine chromosomes, and we see absolutely no evidence for hotspots, this virus integrates randomly in the genome.
Sorry to interrupt you again. Maybe I missed. So in the picture you demonstrated no evidence for mutagenesis over 2 years. Where did you look at for the mutagenesis?
Sorry?
Where did you look at the mutagenesis for the mutagenesis in particular genes or randomly?
Okay, so we looked both at random integration, and then what we did was from the databases, we took the top 10 genes that are linked to cancer in the lung, and we looked to see whether there were any hotspots that favored clonal expansion in or around those top 10 lung genes. And the answer is there is no clonal expansion around that.
All right, thank you.
The final thing is you've got to make enough of this virus, and we started at about 100 microliter scale, and working with the company that I will show you in a minute, we are now at 200 liters scale, and that is sufficient to make enough material to nebulize into our patients. So the next steps with this are we identified the optimal promoter and enhancer combination for the lentivirus, which, if you are interested, is still the human CMV enhancer with an elongation factor 1 alpha promoter. We checked that we can hit the relevant number of cells which we need for clinical benefit, and that number in a recessive condition is about 15%. I showed you the function of the transgene. We needed some money, so we went to the Wellcome Trust, and they gave us £3 million for the first-in-man clinical trial. And then at the same time, big pharmaceutical companies knocked our door down for this product. And in 2018, we signed a tripartite deal with Boehringer Ingelheim and also with the world's best lentiviral manufacturing company, Oxford Biomedica. And over the last 3 years, we have been working with them. And because they liked the data we were generating, there was a press release came out about 6 months ago where Boehringer Ingelheim bought the lentivirus from us. So this is no longer belongs to the Gene Therapy Consortium. This product belongs to Boehringer Ingelheim, and we are working with them in partnership to move as quickly as possible into the clinic. Now, I think we've got about an hour for this talk, so I'm just going to skip over these other slides just for a minute and just go on to some ideas that we've got for other lung diseases. But are there any other questions on cystic fibrosis?
Sorusu olan var mı? Arkadaşlar Türkçe de sorabilirsiniz. Ben yardımcı olabilirim. Sorabilirsek de iyi olur lütfen. Nazlı. So I think there is no question, but I was wondering why didn't you use or try the lentiviral lentiviral vectors to deliver the gene, but you explained that you generate a recombinant virus.
Yes, because the ordinary lentivirus, the proteins on an ordinary lentivirus do not recognize receptors on the airway epithelium. Yeah. And therefore, if you use a standard GP120 lentivirus, nothing will happen because it cannot get into the cell. So you have to pseudotype it with the proteins from Sendai to make it get into the cell and deliver the cargo.
Yeah, that's the brilliant idea. And I was wondering if any of the trials, did you observe inhibitors against the protein or I don't know. So, if you would mention it. But
okay. We have not yet done a single trial with the lentivirus. That is coming up. But when in our 7 clinical trials that we've done with the CFTR protein with the liposome, we have never seen antibodies against the CFTR protein itself. It's a really good question.
Thank you. Please
so
go on. Yeah, thank you.
Yeah, please.
I think there's no question from the audience, so we can continue.
Okay, so one of the things that occur to us is that CFTR is a membrane protein. Is this lentivirus here any good at producing secreted proteins? Because if it is, there are a number of rare genetic lung conditions shown here, which we'll talk about in a minute, which might be amenable to using our lentivirus to secrete proteins into the lung. There are also some commoner lung conditions, and you were mentioning about factor VIII and haemophilia. one of the things that we have shown is if you put the factor 8 gene into this lentivirus, put the lentivirus into the lung of mice, we can secrete enough proteins into the circulation to be therapeutic to treat factor 8 and haemophilia. So it is a very powerful system for secreting proteins. And because of that, we went back to the Wellcome Trust and they gave us just over £6 million to think about other diseases than cystic fibrosis. Now, the reason this vector is so good at secreting proteins is it appears to target every single type of lung cell that we are aware of. So here, for example, is the type 1 alveolar cell here shown in green, which has been stained with GFP from our virus. We proved it was a type 1 cell because we put on a cell-specific antibody, and here is the double staining. And we went through that with type 1, type 2 cells, ciliated cells, so here is gene transduction in the cell. We stained it with tubulin, and then we double stained it. And so we can prove that we got gene transfer in a ciliated cell. And we did it with club cells, goblet cells, basal cells in the airway. And the reason I'm showing you this is just to try and persuade you this vector is very efficient at transducing all the different cell types, and therefore it produces a lot of protein because it doesn't matter which cell type it hits. It hits all the cell types in the lung. Now, I will just very quickly show you 3 examples of other diseases. There is a genetic disease that you're probably aware of called alpha-1 antitrypsin deficiency. And normally, if any of you smoke, and I'm sure you don't, then what you do is you release trypsin, and trypsin destroys the lungs. And this is one of the problems in smoking. Now, we all have an antidote. We have alpha-1 antitrypsin, which stops the trypsin destroying the lung. But these poor patients are born with a genetic deficiency where they do not produce the antitrypsin, and therefore the trypsin destroys the lung. So the obvious thing we need to do to treat these patients is to put the alpha-1 antitrypsin gene into our virus, put it into the lung, and then see if we can stop the trypsin destroying the lung. So what we did was we put human alpha-1 antitrypsin into our virus, and the more you give, the more we produce. But the absolute key question is, what is the clinical benchmark? How much do you need to make a clinical difference? And at the Brompton, we have lots of different mutations of patients with alpha-1 antitrypsin deficiency, and we know that if you have the SZ genotype, then that particular mutation protects you against lung disease. So here is our clinical benchmark of how much we need, and I think I can show you that we can produce that amount. And a single dose will last out for, here in the mouse, for at least 600 days, for 2 years in this mouse model. So we are now spinning out a company around trying to produce a clinical treatment for alpha-1 antitrypsin deficiency. There is another awful disease where patients drown in their own surfactant. So you will know that your lungs cannot expand unless they have the lipid material surfactant in them. And you have to remove that surfactant. And the way you do that is that macrophages come in and eat the surfactant. Macrophages will not eat surfactant unless they have the protein GM-CSF. And what happens in these patients is they have autoantibodies against GM-CSF. So the GM-CSF does not activate the macrophage does not eat the surfactant, and these patients die because they drown in their own surfactant. And the only treatment available that we have at the Brompton is every 6 months they come in, they have a general anaesthetic, we put a tube into their lungs, and we pour a litre of saline into their lung, and we wash out the surfactant. This is the second litre of saline. This is the third litre of saline. And eventually you can wash out the surfactant that is not being eaten by the macrophages. They then go home, and after 6 months they come in again, they have a general anaesthetic, and we repeat this all over again. Now, there is a mouse model of alveolar proteinosis where you can see this is the normal surfactant, and here is the alveolar proteinosis, much like in patients. So what we need to do is we need to put the GM-CSF protein into the lentivirus and put it down into the lungs of these patients. And what we've done in the mouse model is we have put the GM-CSF into the lentivirus. This is the amount of turbidity, this thick material, that is present without the gene therapy. And as we put more and more in, I think you can see we can clear the alveolar proteinosis. So one of the things we are pushing forward quite quickly is to see whether we can produce the gene therapy using GM-CSF for this disease, alveolar proteinosis. And the last example I want to give you is that we can produce now antibodies in the lung using this lentivirus. So here are some mice that have been treated with 10 times the lethal dose of flu. And these mice have to be killed at this point because they lose 20% or more of their body weight. What we then did was put in the lentivirus a gene saying make anti-flu antibodies. And I think you can see that as they lose weight and we treat them, we can recover these mice. So we now have a system where we can produce COVID antibodies, flu antibodies, RSV antibodies, rituximab antibodies in the lungs using this gene therapy vector. So in the last couple of minutes of the talk, I just want to reflect, if I may. I've spent 35 years in this field now. What have we learned as a big strategic picture from our work? Now, I'm going to be controversial. I understand the penalty for being controversial. Now, I think that the most important lesson, and if you take away nothing from this talk, is that gene therapy remains about delivery, delivery, and delivery. And I think if this person, if we were listening to his wise words, he would say that evolution has made the conducting airways to conduct air from the mouth to the alveolus. And what it has not done is to evolve into an absorbing structure, and it is extraordinarily well defended. And I think that Darwin would say, this is going to be very difficult. What is it you are trying to do here? You are trying to work against evolution. Now, we are very, very jealous of the gene therapists who are able to do gene therapy topically. So, for example, you will know that the bone marrow people can remove some bone marrow from the leg. They can do gene therapy in the laboratory. They can put the bone marrow back into the patients, and it is very successful. You will know that there is very good eye gene therapy because you can inject directly into the eye. Very good liver gene therapy because you can inject directly into the liver. Nevertheless, I hope I have been able to persuade you that after 30 years, we now have a vector that can also be quite good in the lungs. But whatever you do, always be concerned whether you can re-deliver your vector, because clinically there is absolutely no point in using a vector that you cannot re-deliver repeatedly. The second thing I think I've learned is that every time you take out a newspaper or a magazine, there is always a new toy, a new technology. So if I ask Google Maps how to go from London to Oxford, it says to me, take a car and you will get there in 1 hour. If I say to Google Maps, I've got this fantastic new technology, how long will it take me to go from London to Oxford with a space shuttle? It will probably say 3 seconds. But maybe on mature reflection, a space shuttle is not the best way to go from London to Oxford. And I would just encourage us to not always think that the new toy is always better than the old toy. The third thing I think we've learned is that all of us in this room, I think, would like to minimize the use of animal models in research. And you will know there are lots and lots of CF mice. None of those CF mice develop lung disease. They're not like patients. And that is because they have modifier genes. They have alternative chloride channels which secrete chloride into the airways in place of the CFTR missing chloride. So there is now a fantastic literature on murine modifier genes in the airways. We also have CF pigs, and these pigs have the most horrendous gut disease. Their guts are completely blocked by mucus. We now have surgeons who have opened pig intensive care units to try and treat these pigs and keep them alive. And we have the most fantastic literature on pig CF ITUs. I just wonder sometimes whether animal models actually slow the translational progress of gene therapy into people. And as long as it is safe to do so, I wonder whether we should move more quickly and more often into human trials rather than spending years and years on animal models. One of the other big issues is manufacturing. Most of the gene therapy that I'm aware of is driven by academics and not industry. And academics have always got to get through this valley of death. Industry will not buy products from academics unless you have gone through this valley of death and shown that it is successful. That means that academics have to pay for the gene therapy materials, which are very expensive. And one of the things that I think we need to do strategically is to accelerate low-cost access for academics to manufacturing of gene therapy materials so that the academic can get across this valley of death and sell to pharmaceutical companies. And we are doing this at scale now in the United Kingdom. One of the things you mentioned earlier, right at the beginning, was the cost of goods. And you will know that this drug, Glybera, which treats lipoprotein lipase deficiency, costs $1.4 million for one dose. This is cure. This is a curative therapy. It really, really works. And the company only sold one dose because the health services cannot afford 1.4 million for a single dose of gene therapy. You will know there is the drug Strimvelis, manufactured through GSK. Its price tag was $700,000, and GSK sold this product, which works brilliantly and is a cure because nobody was buying it from GSK because it is too expensive. And I think we have to be really, really careful about health economics when we devise all this fancy gene therapy. And finally, I would just say to you, after 35 years, we find this very difficult. We don't find gene therapy easy to do. It is high-hanging fruit. We find that it is really, really important to have big teams. We have between 50 and 70 people working on this. We find it is really, really important to be collaborating with industry at the beginning and not at the end of the process. And it is absolutely critical there is international cooperation. And this is all going to go horribly wrong if we end up doing this sort of thing. So my last slide then, just to show you what happens in the UK. These are loads and loads of really, really enthusiastic stakeholders who all want to make sure the United Kingdom is ready to deliver advanced therapy medicinal products, cell and gene therapy. The problem that we found is they need coordinating. It's like herding cats. Everybody was very enthusiastic, but nobody was talking to each other. And so one of the things we did was the National Institute for Health Research, which is our governing body. I brought together a coordinating group where the detail doesn't matter. Everybody who is interested in gene therapy comes together every 3 months, and all we simply do is that we try and reduce the competition and the duplication and just say, if this group is doing something, please, this group, don't duplicate it and don't compete with each other. And we have 5 working groups, which includes training all the research nurses, the doctors. We have a pharmacy training group on how to handle gene therapy. We work with industry. We have all the infrastructure, so you need cubicles to deliver this nebulized gene therapy, for example, and we have the patient and public involvement very much at the center of what we do. Now, I'm sure you're familiar that all new treatments always go through this pattern. First of all, it's science fiction. And then the second thing is every time you go to your doctor, this will cure everything. And then after a short while, you end up with this thing that it actually is completely useless. And then after another short while, you find out that actually it is good treatment for certain diseases. And I think gene therapy is coming to the age where it is good treatment for some diseases, but it has taken us nearly 30 years to fulfill its potential. It is not like small molecules, which take 15 years. Gene therapy is really complicated. So I just want to thank everybody in the consortium that I work with, all the different funders, and this is our website if you're interested in any more detail. So thank you very much for your attention.
I thank you very much. I am really delighted to listen to your talk. It is very informative and very up to date. And there was a question regarding the use of CRISPR-R technology. What would you think of using CRISPR-R technology to treat CF patients?
Exactly. So really, really good question. And this is my reference to the new toy, because CRISPR and gene editing is the most fantastic new toy ever made. It's wonderful, isn't it?
Yeah.
So everybody says to us, why don't you do gene editing and sort that out in the lung? Please beware. You have to get the guide RNA and the CRISPR-Cas into the lung epithelial cells. Well, that's the same problem that we have. We have to get the gene replacement in. So
Yes.
what is the advantage? Now, the next advantage, one of you will say, is if you gene edited, then surely you only have to do it once, because then you have edited it and put it back to normal. That's only true if you edit it in the stem cell.
That's true.
We cannot reach the stem cells in the lung. They are hidden. They are underneath.
Mm-hmm.
And the other thing you will all know is that the CRISPR-Cas and other systems work particularly well with dividing cells. The lung epithelium is not dividing.
Yeah.
The fourth thing you will know is there are off-target effects of the gene editing. It's not that specific.
Mm-hmm.
So for those four reasons, I would really, really encourage you not to think that gene editing is the answer to everything. It's the answer to making cell lines. It's the answer to making animal models. It's the answer to all sorts of fantastic things. It's not the answer to everything.
That's true. I agree with you.
And the trouble is that people put money into the new toy and they take money away from the old toy. Let the old toy have a chance to make its mark.
Yeah. And there's accumulation of like 30 years of information. Like, why not use these information and start a new toy?
Yeah, exactly. It was very encouraging for us to learn that a big company like Boehringer Ingelheim are willing to put a whole load of money. This is not going to be a cheap clinical trial, a whole load of money into doing this for CF. And as I said, we are just spinning out a company for the non-CF indications like the alpha-1 antitrypsin and the alveolar proteinosis, etc.
Yeah, it is good for you that they put that money. Otherwise, it wouldn't be possible. And I was happy to hear that these methods you developed may be a cure to treat the COVID-19 patients. Did I misunderstand you?
So we don't like the word cure. So one of the problems, can I turn the question back to you?
Yes,
Would
sure.
you use our virus to treat COVID patients? And the answer must be no. And it must be no because the cost of a vaccine, at least in the UK, is probably about £3 an injection.
Yeah.
The cost of our lentivirus will be much, much more than that. And it comes down to economics and the cost of goods. Now, of course, you well know that what we are doing with the COVID vaccines is we are putting in the antigen and the patient is making the antibody. You will know that there are immunosenescent patients. We have 500,000 people in Britain who cannot make antibodies when you put the antigen in because they are too old or they are immunocompromised. So for those 500,000 patients, we need passive immunity. We need to put the antibodies into the patient, not ask them to make the antibody. So our system would be perfect for those 500,000 patients. I cannot believe that the National Health Service will pay whatever it's going to be, many tens of thousands of pounds for our virus compared to £3 for a standard Pfizer vaccine.
That's true. I know. Okay, any questions? Any question from audience?
Please challenge me. One of the questions you should ask me is, why did we use saline as the placebo?
Yeah, that's true. I was going to ask you, why did you use saline?
A completely stupid thing to do. When we did our lipid trial, what is the obvious placebo? Well, the first obvious thing to say is, if the gene is complex with the lipid, why don't you just put the lipid in? Mm-hmm. So the lipid, we discovered, had its own pathological effects, which were different to the gene and the lipid together. So it would have given us a false picture. So the right control is what? Let me turn the question back to the audience. What is the correct control?
So what is the correct control, would you think?
Remember, the active is the CF gene with the lipid. What is the pure? You're all brilliant scientists. What is the correct control?
Any answer?
Come on, you know the answer.
They are too shy.
Okay. The correct answer is you should mutate the CF gene so it's not functional, and then put it together with the lipid. So the active is the unmutated CF gene, and the control is the mutated CF gene with the lipid, because when you put that in, it's exactly the same, but it can't produce the CFTR protein.
Yeah.
Why did we not do that? The referees asked us why we didn't do that. The cost of putting in the placebo with a mutated gene was £6 million. I don't have £6 million. I have £6 million for the active treatment. I cannot afford another £6 million for the placebo. So it shouldn't be the lipid, because that's not the right answer. So the only thing left was saline. So when we sent the paper to the New England Journal of Medicine, they turned it down because they thought we had the wrong placebo. The Lancet did take the paper because they understood the issue. But hey ho, it comes down to money in the end.
Yeah, that's true. We have to be concerned about the money.
Dave Fraser, any other questions at all?
Yeah, any questions, please? As I said, you can ask in Turkish, and I can help you. Nazlı, you look like you have a question.
Actually, I'm confused about a little bit about trypsin. Okay.
That's better. You're confused about trypsin?
Is it right now?
Yep, it's fine.
Okay. I have a little big question, because I'm confused about trypsin. You said that trypsin destroy lungs. What did you mean with the sentence? I'm confused about that. Thank you.
Okay. So if you were to smoke a cigarette, and I'm sure you don't, then what happens is the cigarette smoke activates trypsin, the enzyme. And it does that because the trypsin is trying to get rid of the bad things in the cigarette smoke. The trypsin not only gets rid of the bad things in the cigarette smoke, but it destroys the wall of the alveolus at the end, the gas exchanging bit. And so every time you are smoking a cigarette, some of the material is beginning to destroy the lung. And that's why people get COPD, they get bronchitis, they get emphysema. Now, we have a mechanism to block the effect of the trypsin, to stop it. And that is called alpha-1 antitrypsin. It is a protein that if you smoke a cigarette now, your lungs will produce trypsin to defend you, but you must put an end to the trypsin activation, otherwise it destroys the lung. So your body switches on alpha-1 antitrypsin. The people with the genetic disease alpha-1 antitrypsin deficiency can't produce alpha-1 antitrypsin. So when you smoke, the trypsin carries on and carries on and carries on working and destroys more and more of your lung because there is no antitrypsin to stop it.
I think it is clear for Nazlı.
Is that clear?
Yeah.
Thank you. And I'm wondering that how did you handle it? I mean, protein.
How did we produce the alpha-1 antitrypsin? Is that the question?
Yes.
Yes.
This is the
Okay.
question.
So what we did was we put the gene for alpha-1 antitrypsin into our lentivirus, put the lentivirus down into the lungs, and then the virus will transcribe and translate the alpha-1 antitrypsin, which will be secreted into the lung. It then meets the trypsin and neutralizes it. Did that make sense?
Açma istersen.
Yes, Professor. Thank you.
So, any other questions? So I think there's no question, and it was very clear. Like you explained everything you need to explain when you are talking. So I think there's no question left for the audience. It was really very beneficial webinar for me. And I thank you very much again for accepting our invitation. I hope in the very near future, I will host you here in face-to-face meeting.
That will be lovely. Can I say thank you very much indeed? It's been a great pleasure. I'm sorry I wasn't there in person, but, you know, difficult times. But thank you very much. I really enjoyed it. And thanks very much for the invitation again. Oh, there's another question.
Yeah, there's one question.
Are we working with the antibodies? What was the question? Are we working with the
antibodies or antivirus?
Okay, so I can see several. So we are working with the antibodies, but not for COVID at the moment. And we are looking to work with them for anti-flu or for the anti-respiratory syncytial virus, RSV. That's what we're looking for. I think that one of the things that might be really interesting is to make anti-rheumatoid drugs. There are monoclonal antibodies I'm sure you're aware of, like infliximab and rituximab, and we can produce those at large quantities in the lung. So we are working on them. I think the problem is the cost of goods. It really is. At the moment, the virus is expensive. I think it's going to cost 50,000 put a virus into a patient per patient. It's a lot of money. So I think we are working at it, but I think it is quite expensive. There's some other questions there. Can we use lung stem cells as a delivery system after editing with CRISPR-9? So this is now a really exciting question, where I think what you're saying is take some lung stem cells out. So the first problem I would challenge the person who's got the question is, how would you take the lung stem cells out? They are really, really, really well hidden. I don't know how to take them out. Even if you could then, what you're suggesting is CRISPR edit them ex vivo and then put them back in. As soon as you put these cells back in, they will be destroyed by the lung because there is no evolutionary advantage for the cells to take up the niche back in the lungs. So I would challenge back that there are at least two problems with your really elegant idea. So that I think is a really good question. There was also an original question there, which is, why do you repeat the virus treatment every month? No, we were repeating the non-viral treatment every month because it does not integrate into the genome, and therefore it'll only last a few weeks. With the virus, I don't know how often to repeat it. I'm hoping in the patients it's going to be between every 6 and 12 months because it is the lifespan of the respiratory epithelium. So your very good question is entirely dependent on whether we have the knowledge of how long a human respiratory epithelial cell lasts for. And I've asked every world expert that I know, and nobody knows the answer, but people think it is between 6 and 12 months. If the respiratory epithelium only lasts for 3 days, we will have to repeat this every 3 days. If it lasts for 5 years, we'll repeat it every 5 years.
Yeah, thank you very much. I cannot stop asking you a question, but can I have a last question? Eric, how were you sure that you transfect or you delivered the gene only to the lung cells, not the other cells?
Ah, very good. So are you worried that we are going to transduce, say, things in the circulation, the bloodstream, and then we might transduce the ovaries or the testes? Yeah, no, really good. And we are just about to go to the regulators, the MHRA, like the FDA. We're just about to go to them, and they will ask us your question. So the virus is not tropic to the lung. If the virus reaches the bloodstream, it will go to some of those other organs as well. Yeah. We've done some mouse model experiments, and at the moment we have tested every organ in the body, and we can see no evidence that it comes out from the lungs and goes to those other organs. But you are absolutely right, and I hope you will not be on the FDA panel, because you are absolutely right that if it goes into the bloodstream, it will transduce other organs.
Yeah. I wish you all the best then. It won't go.
Well, thank you very much indeed again. It was a great pleasure talking to you all, and thanks so much for the invitation.
Thank you very much. It was very delighted.
Cheerio. Bye-bye.
Evet arkadaşlar, webinarımız bitti. Katılımcıların hepsine ayrı ayrı çok teşekkür ederim.